I'm Russ Jaffe, an internist physician, clinical pathologist, biochemist, certified clinical nutritionist, and I maintain a fellow status in a variety of medical societies, including the American Society for Clinical Pathology, American College of Nutrition, American College of Allergy, Asthma, and Immunology, the Federation of Clinical Immunology Societies, the American Medical Laboratory Immunologist, and I'm an overseas fellow of the Royal Society of Medicine. I'm a senior fellow of the Health Studies Collegium Research Foundation, and I'm director and founder of ELISA Act Biotechnologies and PERQUE. We have some learning objectives, and they are to assess digestive health in the immune system, the role and the mechanisms of food sensitivities. Then we'll look at celiac disease, diagnosis, gluten and non-gluten sensitivity versus intolerance, and its integrative management, with a focus on removing obstacles to recovery and using a physiology-first approach, the alkaline way. Well, let's look together at digestive health from a high level. It's about the conversion of food into energy. So, eat what you can assimilate, what you can utilize, what you can eliminate. Eat what you can take in without burden and harm. Utilize it efficiently to energize and rebuild yourself and keep you resilient. And eliminate the wastes before they accumulate inside you. Now, digestive health does relate to the microbiome in our gut, and it also relates to the microbiome of the soil. So there's a little book, Soil in Your Health. It's written by Beatrice Trum-Hunter, and I'd recommend anything that she's written as accessible, written in a very readable style, yet meticulously researched, documented, and presented in a integrative systems approach, so that you really see the relationships as well as the trees. So you can get back to see the forest and then get in to see the trees, and farther down into the roots of the trees, and farther down into the microbiome that makes the roots, that makes the tree possible. So eat what you can assimilate, utilize, and eliminate. Now, there's a digestive epidemic of epidemics. There are some common complaints, and we could review those briefly. And this epidemic of digestive epidemics, this epidemic of epidemics, was something I learned about as a student in the 1960s and 70s. The common complaints you see on the left: heartburn, abdominal pain, gas and bloating, constipation, diarrhea, or alternating some combination of that. What people kind of call ajna. Then there are medical comorbidities: IBS, irritable bowel syndrome, diverticulitis, diverticulosis, these outpouchings that can fester under certain conditions, and Crohn's disease, ulcerative colitis. You don't want to have an ulcer in your colon. You don't want to have ulcerative colitis. And then there are the silent digestive epidemics, the enteropathy and atrophy of your digestive mechanisms, the maldigestion and dysbiosis. Well, let's look at optimal digestion and health and how they're linked. And it comes down to a fairly simple statement: food intolerances affect nearly everyone at some point. That's from the National Institute of Allergy and Infectious Disease at the National Institutes of Health. Now, if you will, let's look very briefly at the healthy immune responses and what a healthy person's immune response does, usually invisibly. There are no allergies. You're tolerant, which means when anything foreign comes in, your body soaks it up, puts it in a recycling center, and makes use of it. It doesn't provoke an immune reaction or any kind of metabolic reaction. A healthy immune response neutralizes infections and allergens. Anything foreign, an allergen or an infectious particle, gets put into a recycling center, broken down to its building blocks, utilized by the body. A remarkable system. Again, no immune reactivity, just healthy first-line phagocytic function. Soak up the foreigners, put them in the recycling lysosome center, break them down to their building blocks, take those building blocks in, metabolize them. Healthy immune responses are also those that repair us from daily damage. So after defense is done, repair can be accomplished. In general, during the day, the immune system is on defense, and if we get restorative sleep, it repairs us at night. And perhaps more important than all, but certainly important, a healthy immune response deletes the cancer cells that people make every day. So we have a surveillance system that after defense and repair is done, the immune system is able to go around and touch every cell in the body, and the few that are abnormal are marked for recycling or for what's called apoptosis. Now, overloaded immune system means chronic illness. An overloaded reactive immune system, a burdened immune system, means somewhere, often multiple places in the body, there's inflammation, swelling and pain that the doctor calls inflammation, due to repair deficits. Inflammation, unmasked, as we have thought deeply about it and rethought it, is really cumulative repair deficits. So inflammation is not a fire to be fought. Inflammation is a cry for help. It's a deficit in repair, and we best address that by removing obstacles to recovery so that repair can be robust and restore itself to an effective, healthier level. An overloaded immune system almost always means an increase in digestive remnants. Seasonal allergies are, of course, important, but they only exist in people who have other sources of immunoburden, most of that from the digestive tract, from maldigestion and digestive problems. And then there's chronic degenerative and autoimmune illnesses. As on most of the slides, there will be a reference on the bottom that will allow you to dive more deeply into these questions or look more deeply into the evidence that supports them. Now let's look at food intolerances. An example of food intolerance is dysbiosis, unhealthy microflora in the gut. There are 10 times more bugs in the gut than cells in our body, and the composition of that microflora is elegantly complex, still not entirely known, and subject to imbalance that leads to dysbiosis. And dysbiosis and maldigestion are cousins of one another. So if you see this list of bullets, you start at dysbiosis, you end at maldigestion, and in between you have lactose intolerance or malabsorption due to a lactose intolerance, that is, an acquired lactase deficit, a deficiency of the enzyme lactase. Healthy people produce lactase, especially when they're exposed to lactose. However, it's an elective protective enzyme. It takes a lot of energy for the body to produce it, and when the body has to hunker down because it lacks nutrients, it's too acid at the cellular level, or there's too much distress at the hormonal neurochemical level, then the body says, gee, I'm not going to devote the energy to making lactase. I'm really needing to hunker down into survival mode, and when we get back into healthy or thrival or optimum mode, then we'll start electively producing lactase. So most lactose intolerance is acquired in adulthood. It's very rare actually in childhood, but it's acquired in adulthood as an epigenetic downregulation due to lack of good stuff or too much bad stuff. Lack of essential nutrients, lack of essential input, or too much distress hormone, distress neurochemical, or xenotoxin, environmental chemical. Food intolerances extend into the autoimmune celiac disease, but also to just maldigestion-associated gluten intolerance. Gluten is a hard-to-digest substance. It's also what makes bread and grains sticky after cooking. And gluten intolerance might mean that you have celiac disease, but it equally could mean something quite different. It could mean that you're having trouble digesting gluten, and you need to avoid it and restore competence to digestion. Bring back healthy digestion. And then you'll be able to eat bread again. You won't have a lifetime of careful and meticulous avoidance of gluten, which is what celiac disease true sufferers do need to do, along with a health promotion comprehensive program. So just meticulous avoidance of gluten for either those who are gluten intolerant or those with celiac disease is not enough, but it's a good start. Food intolerances can express as the most common adult immunodeficiency. That's IgA deficit or secretory IgA deficit. This again is an epigenetic downregulation of elective protective secretory IgA or systemic IgA, and is another contributor to etiologic or causal contributor to maldigestion. So from dysbiosis to maldigestion, there are many causes of food intolerance. I'm going to help you distinguish them at a clinical level, but also at a mechanism level in the next few minutes. Back in the 1960s again, delayed hypersensitivity was defined. It's types 2, 3, and 4 immune reactions. And if you look at the top left, you see type 2 antibodies. These are it only means the complement fixing, the provocative IgG, IgM, and IgA. Professor Gell and Professor Coombs knew that there were helpful antibodies, like the antibodies we get when we recover from a stress or when we recover from an illness. Those are neutralizing and protective. They weren't talking about that. They were only talking about the harmful type 2 antibodies, the complement-fixing provocative IgG, IgM, or IgA. But then they distinguished another category, immune complexes, type 3 immune complexes, IC. Type 3 immune complexes, IgM, anti-IgG, antigen complexes. The body recognizes that an IgG that's reactive with an antigen stuck to it is even more harmful. And so it wraps an IgM antibody around that complex that's supposed to be removed easily by the spleen and liver when they're healthy, but may not happen. And when it doesn't happen, then the immune complex diseases like vasculitis can occur. Then there's type 4. The type 4 turn out to be direct T-cell responses, no antibodies needed, thank you. And T-cell mediated delayed responses are an important aspect of immune functionality. Now, to keep this sweet and short as best I can, T cells and macrophages release cell signaling interleukin cytokine communication molecules that start immune cascades that lead to inflammation, which is really repair deficits, and comorbid conditions, those linked with the problem. Now let's see what this means in practice and to reinforce the way in which you can now comprehensively, individually, in a very personalized way, use a very strong evidence base for identifying delayed food and chemical hypersensitivities. There are many kinds of allergy lab tests. Some relate to type 1 allergies. These are RAST and skin tests, and I won't be talking about that very much. But I will be talking about delayed allergies, the lymphocyte response assay that's needed to tell good from bad. For example, the LRA by ELISA Act covers all delayed pathways. And as you see on the right, there are 3 different antibodies in the type 2 category. And then to the counterclockwise above type 3, you see the immune complex and relative proportion to its contribution to autoimmune disease. And then type 4, the T-cell mediated. And some people would say that T-cells actually contribute more and immune complexes less. But the LRA by ELISA Act measures all. It's an ex vivo functional lymphocyte response assay that's able to measure all delayed allergy responses and pathways concurrently. Because we use autologous serum, that is the plasma, the fluid that the cells live in is plasma. When the blood clots, that's serum. We get the whole blood. We gently remove the red cells by spinning them out, not by chemically altering. And we take the cell-rich plasma. We take the plasma that has all the constituents that flow in your body, just as they do in the body, because the blood is obtained without any activation. So it's an ex vivo procedure that allows us to interrogate these memory white cells, these lymphocyte white blood cells, and find out what you're truly reacting to as opposed to what you're neutral towards. LRA by ELISA Act are cell cultures. A 1-ounce blood sample can now be used for over 490 different cell cultures. The widest array possible is available from our laboratory. We purify our own antigens. We've made our own novel microtiter environment to do the incubation. And when we started this work, it would take 3 weeks to get information on one substance with a 30% variance for 1 ounce of blood. We realized you needed to test for hundreds of substances in complex cases, and so we were able to microminiaturize the procedure, develop this novel environment in which an amplified ELISA reaction is done on the surface of a living cell. That's the ACT part of the ELISA ACT. And that's unique to us and has given us the opportunity to do over 60,000 cases, over 18 million lymphocyte responses for this wide array of foods, food colorings, additives, and preservatives, and the other categories you can see on the slide. Now, managing food reactivities has to do with substituting for reactive foods from LRA by ELISA ACT tests. That's how they're identified, so that you accurately get the functional, personal, and yet evidence-based delayed hypersensitivity triggers. Then we add an alkaline diet, targeted supplements to correct nutritional deficits, bring them up to a high level of sufficiency, not too high, just an adequate level of sufficiency. Mental and physical activities that evoke healing responses, and where you choose or where you elect a rotation diet. There are some integrative self-assessments I'd like to briefly review. The first is the first AMPH, are the cells in the body too acid? Then we'll look at the gastrointestinal transit time, and then finally the Ascorbate or CCleanse. Well, if the human is like any other organism, and we are, then we are acidic in function, yet alkaline by need. So we make more acid in our cells, and that would accumulate if we didn't replenish with buffering minerals and other alkaline-forming items in our diet and supplements. And after 6 hours of rest, the urine equilibrates with the cells in the bladder and kidney, and you're able to use that urine uniquely. After 6 hours of rest, you're able to check the fresh urine. This is something people do at home using this color strip. And if it stays yellow, you're too acid. If it's green, you're okay. If it's blue, you might have catabolic illness where you're cannibalizing your lean muscle and wasting ammonia in the urine. The pH level on the left is something we recommend with a simple chart. Note the date, note the time, note the pH level, and how do you feel, say on a 1 to 10 scale or a 1 to 100 scale. And people will report rather consistently that when they are more alkaline, they are less symptomatic and more productive. And the other is also true. When they're more acidic, they're less productive and they're more symptomatic. Now let's turn to GI transit time. This is how long from consumption to elimination of food. The average in America is from 36 to 96 plus hours. Toxins can then reabsorb. It drains metabolism, so much of the energy the body is used up because of this slowness of transit, and there are chronic and systemic consequences. We do know what a healthy transit time is. It's 12 to 18 hours, and less than 10 hours is too short, where you could have malnutrition or malabsorption or short bowel syndrome. Here's an example of one of the simple self-assessment tools that we at the Health Studies Collegium have put together, how to do a transit time. And you can see that 6 to 12 capsules of charcoal, depending on your weight, with 8 ounces of water taken between meals, and you note the time when you do that. And then you watch for the black crumbly stuff to come out your tush, and you mark the time, and the difference in hours is your transit time. 12 to 18 is the target. Check your own. There are nutrients that enhance outcomes. Prebiotic fibers. I have learned more recently that more unprocessed fiber is really healthier, 40 to 100 grams a day, whereas most Americans today get 5 to 10, maybe 12 at the most. The unprocessed fibers have 80% soluble and 20% insoluble. The soluble helps feed the organisms. The insoluble binds toxins for removal. Probiotics are also needed. For healthy people, 5 to 50 billion CFU or colony-forming units, 5 to 50 billion live bugs, taken either through fermented foods and or through lyophilized or freeze-dried probiotics, such as the ones we recommend, with 10 different strains of active healthy organisms. And if you're recovering from antibiotics or replenishing the gut after distress, maybe 50 to 100 billion CFU per day is needed. And the third, something we've contributed uniquely, is recycled glutamine. Glutamine recycled with PAC, glutamine that is re-energized and available to the gut for repair 10 times on average because of pyridoxal alpha-ketoglutarate unique ability to pick up the amine, put it back on the glutamine, let that glutamine give energy to the repair process of the gut. And so 1.5 to 6 grams a day taken on rising or before bed and or before exercise, always on an empty stomach, gives you the benefit of 10 times more free glutamine. So if you take 1.5 grams of the glutamine with the pack in it, then you get the equivalent benefit of 15 grams of free glutamine. If you take 6 grams of glutamine with pack, you get the functional benefit of 60 grams of free glutamine. Now, if you take that much free glutamine, you might well develop glutamate elevation as an excitoneurotoxin. On the other hand, if you look and do what we recommend with the recycled glutamine, you're in physiologic consumption range of glutamine. There's no way for glutamate to build up as an excitoneurotoxin due to that recycled glutamine. Fiber, soluble to insoluble, 80 to 20 is what unprocessed fiber has. Soluble fiber dissolves in water, as I indicated, to make a gel-like material that feeds and nourishes the probiotic organisms. It also lowers blood cholesterol and lowers blood sugar levels because it slows the uptake of blood sugar. And we've had the privilege of developing a novel meal that has the lowest glycemic load, the lowest impact on blood sugar ever recorded, proven in an outcome study, an original science outcome study, not borrowed from someone else. Now, the insoluble fiber promotes movement of bulk through the digestive system and increases toxin removal, as well as healthy probiotic growth. PERQUE Regularity Guard is what we recommend. Soluble to insoluble fiber, 80 to 20. It's a functional and a tropho-restorative, that is, working with the body to help the body, healthy transit time being 12 to 18 hours. With 7 grams in a scoop, we recommend starting with 1 scoop a day and building up with more scoops added on a weekly basis. So the second week maybe 2 scoops, the third week maybe 3 scoops, until between diet and supplements you're getting at least 40, but not more than 100 grams a day of unprocessed fiber. PERQUE Regularity Guard is a breakthrough in having a combination of the most pure and unprocessed, yet uncontaminated probiotic fibers. Now, this is a slide that you're welcome to look at your leisure. It shows the many impacts of probiotics and their beneficial effects on, if you look straight down, metabolic effects, if you look up to the right, immunomodulation, and if you look to the upper left, how intestinal microbiota are restored through replenishment of daily probiotic intake. We must take in either live organisms from our diet, which most of us don't do much of anymore, but used to be the rule with fermented foods as the healthiest parts of most diets. And if not that, then through freeze-dried bugs that are put into suspended animation until they're rehydrated inside of you for their maximum benefit. And I recommend the human implantable strains. I think humans are nourished best by other human strains, not by strains that are designed for sheep or cows or pigs. So PERQUE Digestive Guard Forté 10, healthy flora, healthy digestion, a probiotic balance that promotes health with 10 more active strains, more active because they're human implantable compared to others. They implant, they don't just pass through, and they protect, and interestingly, they can crowd out pathogens. So my family, including my children and my parents, have long benefited from this protection. And while many others in our party have often gotten food reactions or food poisoning of a mild sort, fortunately we have not, and we believe it's because we have the healthy bugs that crowd out the bad bugs when we get exposed to them. And we do think we get exposed to them. We're just able to crowd them out so rather than have them take over. Now, PERQUE Endura Pack Guard is safer repair and energy. Recycled glutamine is the PERQUE Endura Pack Guard product. You see on the right a nice healthy irregular surface to the lining of the GI tract, and that depends upon the availability of glutamine, but also butyrate, the short-chain fatty acid. And here we have a patented formula that energizes and promotes repair while it also enhances digestive competence and restores digestive competence for people who have lost it. Nutrients do enhance outcomes. There are enzyme buffering minerals that are often deficient. Magnesium is the forgotten fifth electrolyte, according to Dr. Ron Aline, and uptake block can be overcome. This is something we discovered with concurrent intake of choline citrate. So if you take magnesium and choline citrate at the same time, you can get it in through a novel mechanism, and then the choline and the citrate alkalinize and energize the body to keep the magnesium in so it doesn't run out as fast as you take it in. Buffered reduced 100% L-ascorbates we recommend. We also recommend super multivitamins with energizing Krebs salts and cofactors. And when I say super multivitamin, I mean that you can take an entire bottle of, say, a Theragram or a Centrum Advanced or a one-a-day silver. Take the entire bottle and you will get less nutrition than 2 of our PERQUE Lifeguard Super Multi product. Hydroxycobalamin, the physiologically preferred form of B12, but taken up under the tongue before swallowed so that you can get more predictable uptake that goes to the brain first and then to the body. And there's more. There are other nutrients that enhance outcomes. There are beneficial fats. You need a balance of omega-3 and 6. You do need both. But many people are getting 1 omega-3 for every 50 omega-6. So we recommend changing that oil intake by a specific product, a EPA-DHA guard, PERQUE EPA-DHA guard. And after, say, 3 or 6 months of that, then a maintenance formula, which is PERQUE triple EFA, omega-3, 6, 9, and CLA all in one soft gel. Then we recommend enriching the diet in the detoxifying foods. These are the ones rich in sulfur amino acids. There are 5 of them. The 5 detoxifying foods are garlic, onions, ginger, brassica sprouts, that means broccoli sprouts, and onions. Now, 1 or more of those should be a staple in your diet. You don't need all of them all the time, but you need more than condiment sizes of either onions or garlic or brassica sprouts or ginger or eggs. And even though I send them in a different sequence, it's the same 5. Now, coenzyme Q10, along with mixed natural vitamins E, coenzyme Q10, with additional natural tocopherols and tocotrienols, micellized in 100% rice bran oil for 3-fold better uptake and dramatically better function, because you're not only improving the electron shuttle, the CoQ10, but also the membrane protective vitamins E, all 8 forms, because they have different parts of the body respond to very different forms of the vitamins E. Cholecalciferol, micellized with medium-chain triglyceride MCT to maintain a 50 to 80 nanogram per ml level. That's when you do the blood test for 25-hydroxy-D. You maintain a vitamin D level of 50 to 80 nanograms per ml. And for most people, that means more than a few thousand IU a day. But we now have the test, and I think personalized care means taking as much of the healthy vitamin D, such as the one that I'm talking about, to get to the healthy target zone, which is 50 to 80 nanograms per ml. By comparison, most Americans are below 30, and that doubles or more, it may more than double, the all-cause morbidity mortality risk and the cancer risk for that person. The NHANES survey confirms that we are nutritionally marginal too much of the time. 87% of people had 1 or more essential deficit. If you look further down, 58% had 2 or more deficits. Omega-3 to omega-6 was typically 50 to 100 in too many people. And this is against daily value intake, not optimum intake for resilience and repair and resistance, just to avoid deficiency diseases, pellagra and kwashiorkor. So the standard American diet is sad. We often are over-consuming yet undernourishing ourselves. We're taking in too many simple calories that are devoid of nutritional value. We need more wholeness in ourselves and in the foods we choose, but we want to choose the foods that don't provoke immune defense reactions. Too many of us have acquired enteropathy and atrophy of the digestive competence. Medications do tend to reduce healthy probiotic microflora. This includes everything from proton pump inhibitors and H2 blockers to antibiotics. and other elements that either slow or stimulate the digestive tract. It even includes medications that lower blood sugar like metformin. Diets deficient in fiber are a problem. Environmental toxins are often contributors. Restorative sleep is very important, yet until people have a compelling need, they very often are not willing to afford themselves the necessary restorative sleep. And different people have different needs. I don't tend to need as much to maintain my function, but dear Rebecca needs 8 to 10, sometimes 12 hours of sleep to feel and function well, and then she functions very well. And all of this has to do with epigenetic phenotypic modulation, so we're messing with our genes and the expression of the genes, and I believe that this is a true problem that we can address by choice, not by fear. Now, what are those medications that cause or potentiate maldigestion and food reactivities? Well, they're the painkillers, the COX-2 inhibitors that inhibit repair. They're antibiotics that kill microflora that they're sensitive to them, regardless of whether they're good or bad. The histamine antagonists, the H2 blockers, the proton pump inhibitors, they reduce stomach acid and impair B12 and mineral uptake. And metformin, as I said, the most common, I believe, medication used for diabetes management, adult diabetes management, promotes B12 deficiency. 2 of 3 people have chronic illness. Hidden food allergy plays an etiologic role. The CCleanse is something we recommend as an individual assessment. It goes beyond bowel tolerance. It's much more evidence-based to assess antioxidant needs. Healthy people start with 1.5 grams, which is a half a teaspoon every 15 minutes. That's 6 grams an hour. Moderately ill people, twice as much. And chronically ill people, 2 to 4 times as much. Now, if you look at healthy people, and they do a CCleanse for 2 hours, they'll be taking in 12 grams, and that will be more than enough for a healthy person to cleanse. Moderately ill person, 12 grams an hour, in 2 hours, 24 grams, most of them will cleanse. Chronically ill people may need 24 to 48 grams an hour, but even they will mostly cleanse on 48 to 96 grams in 2 hours, and then maybe another third hour, which brings you up well above 100 grams. And if you have a short half-life, if you're consuming ascorbate at a prodigious rate, it's not unusual to have that higher requirement for your CCleanse. The idea here is to ramp up quickly and saturate the body with beneficial ascorbate, fully buffered, fully reduced, all L-form ascorbate, until toxins are pumped out, and you feel better. Now, very often people start, and after 2 or 3 hours, they're still nowhere near their cleanse, and we recommend stopping and coming back the next week at a higher level. And there are certain other nuances that we recommend that you take a look at in the Joy of Food, the Alkaline Way Guide. Here are some exemplary data. On the far left, the yellow healthy people. On the dark blue, the moderately ill or healthy people, the walking ambulatory wounded as they're called. The people with symptoms in the light blue, and the people with profound chronic degenerative or autoimmune persistent and multiple conditions on the far right in red. The yellow healthy group calibrated or had their cleanse in less than 4 grams. The dark blue, 5 to 10 grams. The people with symptoms, 10 to 130 grams, and the folks with multiple autoimmune or chronic degenerative illnesses, between 130 and 200 plus grams. This seems like a lot, but healthy people have a 30-day half-life to ascorbate, and it doesn't take much for them to cleanse. But people with a 30-minute half-life who are using it up so fast, you can easily understand, when you understand that, why it takes so much for them to cleanse. There are also some predictive tests that I want to recommend to you. Predictive lab tests with healthy goal values. High sensitivity C reactive protein should be less than 0.5 for repair deficit inflammation. Homocysteine less than 6 for methylation detoxification. Oxidative stress and antioxidant sufficiency with a goal value of having oxidized LDL, HDL, and 8-oxoguanine near absent. For celiac disease, you do want to start with the serologic antibody static tests. This would be like an EMA-IgA or a TTG or a DGA or an AGA. And if they're positive, then you want to do an LRA by ELISA test to find the true hypersensitivities in contrast to the neutralizing serologic and beneficial antibodies that you need to distinguish by a functional test, the LRA by ELISA Act being such a functional procedure. So immune system to enhance and stop the burden. We have hospitable host to pathogens where tolerance is lost. We want to restore tolerance in both the individual and at the immune cellular level. Allergens do provoke symptoms. When we overload the body's immune system with foreign reactive antigens, then repair is deferred because defense comes first, and only functional tests can really identify the causes of immune overload. And as we've always recommended, being scientists as well as clinicians, we go by results, one patient, one benefit at a time. Now, this physiology first approach has a comprehensive plan to lower the burden on the immune system by substituting systematically and knowledgeably for reactivities, being bioavailable to nutritional supplements, and using bioavailable nutritional supplements adequately, and knowing what is needed and hitting the bioactive biomarker goal targets, and practicing lifelong health the alkaline way is what we recommend, and particularly the Joy of Food, the Alkaline Way Guide. Here's the Alkaline Way chart. This is the food and chemical effects on body acid-alkaline balance. And basically, the more alkaline foods are to the left, the more acid foods to the right. And we recommend you download this online or contact our client services to help you understand how to apply this and help your food be your medicine and your medicine be your food. So the path to health is that most of us give it away, and then we realize that our biological health bank account is overdrawn. We have tenfold reserves to replenish or deplete, and we deplete them. And then sustainable health can come back. Sustainable health can come from just focusing on what we eat and drink, what we think and do, all four, eat and drink, think and do, in a comprehensive, balanced, sensible, but yet personalized way, with reactive and functional predictive biomarkers to know, to reassure us that we're going in the right direction. All of this is what we've synthesized. It's the best of what I've learned over the decades. It's what we call the alkaline way. So paraphrasing Hippocrates, Gell, and Huang Ti and the World Health Organization, health is more than the absence of disease. Health is the fullest physical, mental, and spiritual expression attainable by each individual. We each have our own limits. I believe we should live well and wisely with them and at them. Well, I'm Russ Jaffe. It's been a pleasure to bring you this information. If you enjoy it, please tell your friends. Please look at the archive of other similar discussions. And if you have any suggestions or any critique, please contact me. May we all be well and happy, and thank you for your interest in health and in being well.